Hi everyone. What we've done already today is we've gone ahead and done a plate where we're sitting off to the side here. The enzyme assay is incubating, and we're generating, it's going ahead and generating phosphate from the P and PP. And the next thing to do is to be able to do a protein assay. And you might wonder, why are we doing a protein assay? Well, we want to be able to compare from sample to sample and from experiment to experiment and mutant to wild type. We'll make some assumptions that we want to look at the amount of activity there is. So we have the enzyme activity, but we're going to divide that by the amount of protein present. So like for example, if one culture grew a little bit more than the other culture and there's total more protein there, you might actually see more activity in that sample. But we'll go ahead and divide that by the amount of protein present, and that'll normalize everything. So we'll do that, and we'll be able to compare between days and be able to talk about how the activity of the mutant may or may not be greater or lesser activity than the wild type. So the first step we're going to do here is we're going to go ahead and do the protein assay. And what we're going to do is put the samples on the plate just the same way we did for the enzyme assay. Here we go. What I'll do here is the lysate sample. I will add 20 microliters of the lysate to A1. Then I will add 10 microliters of the lysate to A2. And then we'll go ahead and load 5 microliters to A3. So the lysate's been done. Next, we'll add buffer to those samples to make that dilution. And we'll use that same buffer that we used the last time. So what I'll do here is I'll go to 10 microliters on the pipette. And we'll add 10 microliters to A2. And then we'll add 15 microliters to A3. So now we've gotten our lysate has been diluted, so then there's neat lysate, no dilution, two-fold diluted, and now four-fold diluted. The next step to do is to go ahead and load our samples onto the plate as well. And what I'll do here is, like I did last time, is I will open all the tubes on the row that need to be done. So the first two samples we have here are the load samples. So load 1 goes into A4, and load 2 goes into A5. So I'll go ahead and close those two samples up. Oops, I don't need to do that. Okay, the next step along the way is to do the wash, in which we wash the load material off, make sure everything binds. And then we did 3 steps of elution, a 0.1, a 0.3, and a 0.5, and these are all the 0.1s. So I will go ahead and add those, and they go into B1 to B10. So I just loaded wash number 5 into B5. Now it's tube number 1 from the 0.1 molar elution, so that goes into 6, 9, and 10. Next, I'll go ahead and pop the caps here for these samples for the next row. And so all these go into row C. So 0.3 molar wash, tube number 1 goes into C1, 2 goes into C2, 3 goes into C3, 4 goes into C4, and 5 goes into C5. Next, the 0.6 molar washes. Number 1 goes into C6, 2 goes into 7, 3 goes into 8, 4 goes into 9, and, oops, and 10. Okay, so now I've loaded them all the way across. So on this plate we have the lysate, lysate 2-fold diluted, 4-fold diluted. Then we have wash 1, load 2, and then it's wash 1, 2, 3, 4, 5. 0.1 molar elution, 0.1, 0.2, 0.3, 0.4, 0.5. Then 0.3 molar elution, tube 1, tube 3, tube 4, tube 5. And then the 0.6 molar washes go into C6, C7, C8, C9, and C10. Next we'll have to go ahead and we will do the protein standard. All right. Next we have to make a protein standard. And we've done this before. We did remember the first experiment we did 2 types of serial dilutions. And earlier today we did dilution on the plate, which we took 330 microliters of PNP and then took 110 out and then took 110 and put into several wells in a row. This time what we'll do is we'll make a standard in tubes outside. So the standard concentration of BSA that we have in the solution is 2 milligrams per mil. So what I did is I set up a series of tubes where I labeled 1, 0.5, 0.25, 0.125, 0.0625, and 0.3125 mgs per mil. So they're all in here. So what I'll do here is I will go ahead and add water. Oops. I'll add water to each one of these. What I do to make sure is I just going to lift them all up, make sure that I added. So I have 50 microliters of water in each of these tubes. The next step is to add 50 microliters of 2 mg per mil standard to tube number 1. So I'll take 50 microliters out. I'll add it to this tube, and what I'll do is I'll add 50 microliters and pipette up and down. So I've mixed it. Now next I will take 50 microliters out and add that to the next tube. Notice what I did is I went ahead and picked the tube up. I closed the tube off so I know that I've already taken the sample out of it. And I've taken the 50 microliters out, close that tube. Then I'll pick up the next tube and add 50 microliters. This way is the way that I go ahead and do it to make sure that I haven't made a mistake. Just keep doing that for each of these samples. So this is the 0.0625 sample. Take 50 microliters out. And then we'll add 50 microliters to the last tube. And we won't be withdrawing anything from this one. So what I have now is I have a series of tubes. I have some 2 mg per mil sitting over here. And then I have 1 milligram per mil, 0.5, 0.25, 0.125, 0.0625, 0.03125 mgs per mil. So I have a series of dilutions here. Next, I will go ahead and add these to the wells to create a standard. I just realized that what I did here is I skipped a step from on your protocol. On your protocol, you do the wild type on the plate as well. You do but since I didn't have that, I decided that I wouldn't do that today. I'll only do the top part of the plate. So it's time to go ahead and do the standards here. I will go ahead and get the 2 mg per mil standard, and I will put that in wells D1 and E1. Get rid of the tips, it's a different sample. Now I'll go to the 0.5. I mean the 1 mg per mil. Take 25 microliters out. Put that in D1 and E1. The third one goes into D3, E3, D4, E4. Right, just check and make sure. This is the 5th sample, goes into D5 and E5. D6, E6. And the last sample goes into D7, E7. And next I will go ahead and create a blank by putting water in D8 and E8. So I now I've loaded, now this plate has all the samples to be analyzed in them. So I have all of the column fractions, I have the lysate, the column fractions, and my standards are all on the plate. The next step of the process, and you guys will be hopefully doing BCA assay. Unfortunately, the reagent isn't here right now as I film this, so I'm going to go ahead and use the Bradford reagent. I don't care for it as much. I think the BCA assay is a superior assay, but since we don't have it, we can't use it. Hopefully, you guys will not be using it. So here's the Bradford assay. The reagent looks a little bit light. It's homemade. We'll have to see how it works. So what we'll do is, all the wells that have samples in them, I will add 200 microliters of reagent. So the first row has 5 samples. Second row has 10 samples. That's number 10. And I'll just go ahead and go back this way. Now I'll add the reagent to all the standards. Notice how quickly the reaction occurs? That's the one really nice benefit to this method. We go to number 8, which is here. You guys will be using BCA. BCA basically is this. You do the assay the same way. You're basically adding 200 microliters of the reagent to the well. So this simulation is very good. So what I've now done here. Let's go through this again. The plate has the lysate, 2-fold, 4-fold diluted. The load samples, we have the wash samples, so 1 through 5. We have the 0.1 molar 1 through 5, 0.3 molar 1 through 5, and 0.6 molar 1 through 5. And then we have standards loaded on the plate, 2 mg, 1 mg, 0.5, 0.25, 1.25, 0.0625, 0.3125, and then water and in duplicate here. I don't have the wells rows F, G, and H are empty for me because I only did just the wild type. We do in the experimental, so you'll have equivalent samples on here. It'd be lysate, 2-fold, 4-fold, load 1, load 2, wash 1 to 5, 0.1 molar, elution 1 to 5, 0.3 molar 1 to 5, and 0.6 molar 1 to 5 on your plate. Go ahead, and these need to sit for about 2 to 5 minutes, and then give them to me or the TA, and we'll read them on the plate reader. I will go ahead and create a video on sort of analyzing all this data and normalizing it so you can see what I would do for the graphs. But hopefully this helps, and we will see you later. Bye.